availability first, and then advances regarding culture cells provided several pertinent in vitro tests which are now an integral part of the processes used in evaluating the biocompatibility of implant materials and devices. Lecture 27 is a review of how cultured cell models are used in vitro in the processes of evaluating the biocompatibility of implantable materials and devices. Before we start discussing the use of culture cells, we should be reminded of some very important aspects. Specifically, from biology and physiology, we know how cells interact with substrates. Specifically, cells do not interact with bare material surfaces. The pre-adsorbed proteins are the mediators which are needed to bring about the desirable outcome, namely the adhesive interaction of mammalian cells and, of course, other cells with the particular material surface. We had reviewed in a previous lecture these facts, namely that the proteins which are abundant in body fluids and in various tissues of the body, within seconds from the introduction of materials inside the body, are covered by adsorbed proteins. These proteins mediate the subsequent interactions of cells with substrates and promote cell functions such as adhesion, migration, proliferation, all of these cell functions which are pertinent to new tissue formation. And new tissue formation, of course, is the most desirable outcome in the process of wound healing and the most desirable outcome pertinent to the success of the implantation of our favorite biomaterials and biomedical devices in the body. Additional reminders regarding the role of adsorbed proteins in subsequent cell interactions with biomaterials are the following facts. The biomaterial surface properties do affect and dictate the type, the distribution, and conformation of adsorbed proteins. The adsorbed proteins dictate how subsequently the cells will be recruited, whether they will adhere or not, and whether they will function or not. Interactions of cells with proteins are specific, and of course they involve very well studied these days interactions of proteins, the so-called receptors on cell membranes, with specific and select domains on proteins, and those of course are called, those regions are called epitopes, or sometimes they're referred to as ligands. Studies using culture cells in determining the biocompatibility of implantable materials should be preceded by appropriate tests regarding the interaction of proteins which are specific to the cells under consideration and specific to the tissues in which these cells belong. These protein studies should be conducted and completed before any experiments involving cells. These assays determine the type of adsorbed protein and the amount of the adsorbed protein, and include laboratory techniques such as high-performance liquid chromatography, HPLC, calorimetric assays. What happens in this particular case is to stain appropriately with some stains the proteins adsorbed on the surface of specimens of the biomaterial tested, and then examine under microscopy to see where the color is, therefore where the protein has adsorbed. These days, availability of fluorescent stains was the impetus for the development of fluorescent assays. The stains now can fluoresce, and the examination of where the proteins are adsorbed and distributed on the surface of biomaterial specimens, of course, requires special instrumentation, which are fluorescent microscopes. Other assays used these days is the so-called ELISA, or enzyme-linked immunoabsorbance assay. This is a technique which allows determination of very small amounts of the adsorbed protein, very well considered, a highly considered, I should say, experimental technique. Western blotting, of course, is another assay which is used these days for the determination of adsorbed protein type and concentration. Successful completion of the experiments regarding protein adsorption on biomaterial specimens provides the prerequisites needed to move on to the next step, namely the use of cellular models, culture cells in vitro. How do we choose these mammalian cells? We have to know the tissue which will be surrounding the implantable material and device, and then judiciously choose the predominant cells of that particular tissue, procure them from appropriate companies which sell them, and then use them in studies which will determine how these cells interact with the biomaterial specimens under consideration. This kind of study will give us some very important information regarding how the implant materials will be interacting with the cells of the surrounding tissues. These tests can be conducted using various types of cells because, as we know, tissues contain more than one type of cell, although some cells are always considered the predominant cells of a particular tissue. We can repeat this test using one type of cell at a time, and then compare the results to reach a conclusion of whether this particular materials under consideration are appropriate for the application which we have in mind. After we choose the appropriate representative cell from the surrounding tissues, then specific cell functions for the application should be included in the experiments. The functions of the major cell types are going to be priority. I mentioned that already, but one, if we want to be thorough, we can include the cell types which are not as predominant in a particular tissue if the particular application justifies such type of a study. If we are afraid of contamination with bacteria and therefore with infection, then separate experiments should be conducted using some very characteristic types of bacteria. Keep in mind that use of bacteria cannot be allowed in the same laboratory where you have culture cells, because once bacteria contaminate the mammalian cell cultures or the laboratory, they are very difficult to get rid of them. So, studies using in potential infected microorganisms must always be conducted in separate appropriate laboratories. There are many cell functions which we could consider conducting when we use culture cell models. First and foremost should be tests for viability, which means that the cells must remain alive and functioning after they interact with the new biomaterial specimens. Cell damage or death is not allowed. Other functions will be adhesion of the cells, proliferation, migration and occupation of the substrate surface, definitely synthesis and deposition of proteins pertinent to new tissue formation. If we're using stem cells, if we have phenotype expression and changes, programmed cell death known as apoptosis, and in addition other functions pertinent to specific cells and to specific tissues as needed. Since viability of cells is the first and foremost test that we should conduct, cytotoxicity assays are available and they are now standard techniques in many laboratories. What is done in this particular case is to put the culture cells on the top of the substrate and in what we call direct contact assay determine whether the cells remain viable or they die, or sometimes they may not die, but they may be very badly affected from that particular contact. Another assay is to use the agar diffusion assay. Agar is a polymer which is used extensively in the microbiology laboratories, and what one does is to cut off small circles like little wells and put the cells there, and in adjacent or surrounding, I should say, similar small holes on the agar layer, which now are filled with specimens which of supernatant media collected from the specimens after they've been bathed in those media or buffers. Then examination of whether the contents of those elutions affecting the cells in the central wall, whether the cells again survive, are alive, viable, and functioning. Elution assay is related to the agar diffusion assay. What is done is to bathe specimens of the new material in a buffer like isotonic saline, and then keep these specimens in an environment which simulates the conditions of the human body, for example, temperature 37 degrees, and from time to time take small volumes called aliquots of the supernatant buffer and analyze the contents. Since you start with a buffer which means does not have any other chemical compounds, if there are chemical compounds which are eluted from the specimen, in the case of metals could be ions, in the case of polymers could be monomers, those can be determined in terms of what they are and what is their concentration. Then this particular supernatant medium can be used to expose the cells to this environment. And again, depending on the type of these eluted chemical components and their concentration. Sometimes the cells may or may not survive, may and may not function. Once the cell survival and viability is successfully determined, then subsequent tests regarding the function of cells, functions pertinent to new tissue formation, for example, can be determined by doing various experiments which are now available in appropriate protocols. For example, cell adhesion and spreading assays, cell migration, proliferation assays, and then determining down to the molecular level what is happening in terms of the processes of whether the cells are going to function in terms of proliferation or in terms of the formation and deposition of certain extracellular matrix proteins. These DNA and RNA assays, of course, are the polymerase chain reaction, or PCR, reverse transcription polymerase chain reaction, also known as RT-PCR, southern and northern blotting. Last but not least, immunostaining assays to determine what proteins the cells produce and deposited in their extracellular matrix. The current status regarding culture cell models is the following. These in vitro systems are recognized as very effective screening methods for many types and many specimens of materials considered for implantation. If the experiments using culture cells are not successful, the particular process stops and the biomaterial science and engineers have to go back to the drawing board and start designing and formulating new and different materials. We have to keep in mind that in vitro cell models have certain important limitations. Yes, they provide valuable information on which we as biomedical engineers can make very important decisions. However, the environment in which the culture cells are used in the laboratory does not really duplicate and simulate exactly the complex, multifaceted, interactive, and dynamic events which take place at the tissue biomaterial interaction inside the body. The conclusion for the use of these particular models is that they provide important information, but that information cannot be translated into all kinds of conclusions which are pertinent to what may or may not happen inside the bodies of either animals or humans. For this purpose, additional experiments and different types of materials are being used in the process of evaluating the biocompatibility of implantable materials and devices. This is the end of Lecture 27, Evaluation of Biocompatibility, In Vitro, Part 2, Cultured Cell Models.